to our next speaker, who is Arun Dhanapal from the University of Missouri. And Arun's going to be talking about mining soybean genetic diversity to identify genetic markers and physiological mechanisms underlying carbon isotope discrimination.
Good morning to all of you. I thank the organizing committee chair and co-chair for giving me this wonderful opportunity. I would like to start with the Pasteur's equation, where the grain yield is equal to amount of water used into water use efficiency into harvest index. As you know that the water use efficiency is one of the difficult to measure in the field. So we used carbon isotope discrimination as a surrogate to measure water use efficiency in our soybean study. To our knowledge, genetics of CID in soybean is minimal at the best. So actually we screened large amount of soybean germplasm in order to do GWAS study to identify putative loci. And also another reason is to identify extreme genotypes for making crosses and also to do further physiological studies. So as you can see from this graph, the number of genotypes is on the x-axis and the soybean biomass CID is on the y-axis. You see a good variability for the CID in our collection. So we actually conducted the GWAS analysis over 4 environments with 3 replications, and we identified about 39 SNP markers tagging 21 putative loci. And this is the chromosome map, which was the putative loci for CID, where you can see the chromosomal position on the x-axis and the chromosome on the y-axis. And the dots that constituted the line from 1 to 20 are nothing but the number of SNPs, which is used about 12,000 SNPs, which we used. And the small green circle, blue and red, are nothing but the SNPs that are identified based on the different analysis method. We did report previously identified QTL with the purple squares and the pink and purple triangles. So one of the important things which we need to see in this figure is the genetic loci which are being identified by more than one analysis method, which are being highlighted with the yellow circles over here. So with this, we can rightly say that the good variability for CID existed in the soybean germplasm that we studied, and it can be rightly used in the GWAS approach to identify genetic loci. Once we identified the extreme, our next interest was to see how the soybean CID is related with the water use efficiency. So in order to find this, we actually done both field and greenhouse experiment. All the field experiments was done in the drought simulator or rainout center that we had at the Bradford Research Farm at University of Missouri, and the greenhouse we used was the Ashland greenhouse in University of Missouri as well. So we actually examined the contrasting genotypes for low CID and high CID, imposing different water availability treatments and calculated their water use. And later we determined the carbon isotope signatures for all the genotypes. As we selected the CID, as we selected the genotypes based on the CID, here you can see how they perform. I mean, these two lines which are with the red bars are the lines which are selected for the low CID, which showed the higher water use efficiency. And these two lines are for the higher CID, which showed low water use efficiency. And the graph on the right, which shows, which is for like a check for us to see whether what we have done is right. So these genotypes under water limited condition showed higher water use efficiency, which is what is important for us. So we were thinking about whether we could still see some differences using some high-throughput approaches. So we actually determined the high-throughput thermal imaging for canopy temperature depression. And what we found out was the low CID genotypes had lower canopy temperature depression, and as the high CID genotypes had higher CTD. So the correlation between the CTD and the CID was positive, whereas the correlation between CTD and water use efficiency was negative. And this result shows us that thermal images can be successfully used in order to differentiate the genotypes for water use efficiency. And these results are coming from the pot experiments which was conducted in the hoop house, where we were interested to see how these extreme genotypes respond to the photosynthesis and stomatal conductance. As in the x-axis, you see the time of the day, and the photosynthesis on the y-axis on the left over here, where you see the high CID genotypes under well water condition, it showed higher photosynthesis. And same, the high CID also had the higher stomatal conductance. So when we say that there is higher stomatal conductance, there should be higher transpiration. As you can see in the figure over here, we did observe that as well. I mean, the transpiration was actually calculated by placing the pots on the scale and measuring the water loss for every 5 minutes. And As you can see here, the high CAD genotypes had higher transpiration, and when we try to average them, and the sum of 3 days transpiration is shown over here, where the high CAD genotypes show higher transpiration. So in conclusion, we can say that the high CAD genotypes have higher stomatal conductance, photosynthetic rate, and transpiration rate. So as you can see from these results, you may be assuming whether whether low CAD genotypes are more resistant to yield. I mean, that is what we actually did some preliminary experiment, and I have some results over here, where you can see we have done two type of experiments, one using the plant introduction and one using elect cultivars. So the whole CAD on the x-axis over here, and the yield for the plant introduction on the y-axis, where you see a good relationship between this. But I think this is not our interest, because when you select for a higher yield over here, then you will be selecting for the higher CAD. Actually, we need to select for the low CAD with the higher yield. So I mean, in the figure on the right, where you can see with the elite cultivars, the four elite cultivars which were cultivated in 4 different locations, we see a good negative relationship over here, where when you see here for the low CAD, we are selecting a higher yield. This is what is more interesting for us. And to answer the question whether the high yield is associated with the higher water use efficiency, of course, yes, based on the allied cultivars. Of course, the difference exists between the cultivars and the PA. And one of the reasons for this would be that the PA are from a germplasm bank, or they are always low yielding, where the allied cultivars, there is a good yield. So we need to study this further. And another reason might be also the effect of the environmental condition, where these two experiments was not conducted in the same year. They have been conducted in the different year. So with that, I would like to conclude that the genetic variability for the CAD exists in the soybean genotypes, and the genotypes with the lower CAD exhibited higher water use efficiency in both in our greenhouse studies and field studies. And thermal imaging can be useful to identify the genotypes differing for the water use efficiency. So the superior water use efficiency may be leveraged to develop drought-tolerant cultivars. So our future work would involve confirming these genetic markers for use in selecting the genotype for higher water use efficiency. And we would like to further know whether is there any yield penalty or improvement with selection for, is there is any room for improvement for selection with water use efficiency in soybean. So we are more interested also to see non-stomatal limitations such as mesophyll conductance, density of stomata, and also rubisco partitioning may play an important role. So we are trying to explore this further. And we are also interested to see how the genotype into environmental interactions works for our extreme CAD genotypes. With that, I would like to acknowledge my supervisor, Felix Frici, and most of the field work screening has been done by Singh and Valerio, and the greenhouse and the growth chamber experiments was done by Brett Naylor and Alvaro, and we had collaborators from USDA, ARS, and University of Arkansas.
But can we take questions at the end
Yes, sure.
together? Yeah, thanks.